Good morning, everyone, and thank you for being here today. I am pleased to welcome you to this presentation. We will be discussing the risk assessment and validation of 0.2 micron sterilizing filters in pharmaceutical manufacturing. Over the course of this session, we'll explore the key aspects of filter risk assessment, validation processes, and their critical role in ensuring product safety. I look forward to sharing insights and engaging with you on this important topic. Let's start by discussing sterilizing filtration using 0.2 micron filters. This step is absolutely critical in pharmaceutical manufacturing, especially when it comes to heat-sensitive sterile products that can't go through terminal sterilization. Because these products are sensitive to heat, filtration becomes our primary means of ensuring sterility. It's important to emphasize that a scientifically justified risk assessment, along with a robust validation program, is essential. This helps us demonstrate that these sterilizing-grade filters reliably remove microorganisms every time, without negatively impacting the product quality or the overall process performance. Now, moving on to why this step is so important. First, it ensures sterility assurance. That means we can confidently say the product is free from harmful microbes. It also prevents microbial contamination, which is crucial for patient safety. Furthermore, sterilizing filtration supports compliance with aseptic processing regulations, helping us meet strict industry standards. And ultimately, all these efforts are about protecting the patient. Finally, it's worth noting that these practices meet global regulatory expectations, which is vital for bringing products to markets worldwide. So, in our overall discussion, understanding the significance of sterilizing filtration helps us appreciate how it safeguards both product integrity and patient health. When we look at applicable regulatory guidelines, there are several key references that shape our sterilization practices. For instance, in the United States, the FDA's 21 CFR Part 210 and 211 set the standards for good manufacturing practices. In Europe, we have the EU GMP Annex 1, which specifically addresses sterile medicinal products. The USP 1229 provides guidance on sterilizing filtration, emphasizing its critical role in ensuring product safety. Additionally, the PDA Technical Report Number 26 offers detailed recommendations on sterilizing filtration processes, while ISO 13408 focuses on aseptic processing techniques. The WHO GMP guidelines and PIC-S guidance on sterile manufacturing also contribute to the global regulatory landscape, ensuring consistent standards. Lastly, ICH Q9 on quality risk management underscores the importance of assessing and controlling risks throughout the process. Moving to regulatory expectations, it's essential that we utilize validated sterilizing-grade filters to ensure effective sterilization. Adopting a risk-based contamination control strategy helps us proactively minimize contamination risks. Integrity testing and traceability are crucial for verifying filter performance and maintaining full process transparency. Scientific justification of the filtration process underpins our validation efforts, demonstrating that our methods are robust and reliable. And finally, comprehensive documentation and lifecycle management ensure that our sterilization processes remain compliant and effective over time. All of these elements connect back to our overarching goal, delivering safe, high-quality products through adherence to regulatory standards. Let's take a closer look at what a 0.2 micron sterilizing-grade filter actually is. Essentially, it's a filter designed specifically to remove microorganisms and tiny particulate contaminants from pharmaceutical solutions. This level of filtration is critical in ensuring the safety and purity of the final product. Now, where do we typically use these filters? They are essential for injectable products, where sterility is absolutely non-negotiable. They're also used with biologics and vaccines, helping to maintain their integrity and safety. Ophthalmic preparations, which require the highest purity standards, rely heavily on these filters. Additionally, they are employed in sterile bulk solutions and in media and buffer filtration, ensuring that every step of the manufacturing process meets strict cleanliness standards. Moving on to the materials used in these membranes, we have several common options. Polyethersulfone, or PES, is known for its robustness and compatibility with a wide range of solutions. PVDF, or polyvinylidene fluoride, offers excellent chemical resistance and flow rates. Nylon is another popular choice, valued for its durability and filtration efficiency. Lastly, PTFE, or polytetrafluoroethylene, is prized for its high chemical inertness and temperature resistance. Understanding these materials helps us choose the right filter for each application, ensuring we meet both regulatory standards and the specific needs of our products. So, the key takeaway here is that the right membrane material, combined with the 0.2 micron filtration level, provides an effective barrier against contaminants, safeguarding the entire manufacturing process and patient safety. Let's take a closer look at the scientific basis of sterilizing filtration. When we talk about filtration mechanisms, there are a few key processes at work. The first is size exclusion, which means particles are physically blocked because they're larger than the filter's pores. Then, there's adsorption, where contaminants stick to the filter material itself. And finally, tortuous path retention, which involves contaminants getting caught as they navigate a complex, winding path through the filter. Now, for validation purposes, we use a standard organism, Brevandemonas diminuta. This bacterium is small enough to challenge the filter's effectiveness, making it a reliable test organism. From a scientific standpoint, most bacterial contaminants are larger than 0.2 microns, so filters designed to retain these organisms are well-positioned to ensure safety. We validate sterilizing filters by challenging them with bacterial organisms under the worst-case conditions, meaning the most difficult scenarios, to confirm they can reliably retain microbes. The main objective here is to demonstrate that our filtration process can consistently retain microbes during routine manufacturing. This ensures the safety and sterility of our products, which is ultimately our top priority. Let's delve into the regulatory expectations surrounding sterile filtration. As part of this, we apply the principles of ICH-Q9, which focuses on quality risk management. This approach helps us systematically evaluate and control potential risks throughout the process. When assessing risks, we consider several key factors. First, the product characteristics. Understanding the nature of what we're filtering is crucial. Next, we look at the bioburden level, which indicates the microbial load present. Filter compatibility is also important, ensuring the filter material suits the product. We examine process parameters like pressure and flow rate, as these can influence filtration efficiency. The hold time before filtration is another factor, as delays can impact sterility. And finally, environmental conditions during processing, such as temperature and humidity, must be controlled to prevent contamination. Now, in sterile filtration, applying a quality risk management approach means we identify potential risks that could compromise sterility assurance, the overall quality of the product, patient safety, and the reliability of the process. The goal is to implement controls that mitigate these risks effectively, ensuring that every step aligns with regulatory expectations and maintains the highest standards of product integrity. Let's talk about the impact of filtration-related risks. When these risks aren't properly managed, they can lead to a loss of sterility assurance, which is critical for patient safety and product efficacy. If sterility is compromised, it can result in product contamination, posing serious health risks. In worst-case scenarios, contaminated products might need to be rejected or recalled, leading to significant financial and reputational damage. Now, focusing on the common risks associated with filtration, many of these are related to the filtration process itself. For example, filter blockage or fouling can occur when particles or biofilms accumulate, reducing the filter's effectiveness. Filter integrity failure is another major concern, as it can allow contaminants to pass through. Other risks include an excessive bioburden load, which can overwhelm the filtration system, and product adsorption to the membrane, which can reduce yield or alter product quality. Extractables and leachables from the filter materials can also contaminate the product, and pressure fluctuations may cause microtears or compromise filter integrity. Lastly, improper installation or handling of filters can introduce defects or contamination, emphasizing the importance of proper procedures. All these factors highlight why rigorous control and validation of filtration processes are essential to maintain product safety and quality throughout manufacturing. Now, let's discuss the key validation activities involved in ensuring our filtration process meets the necessary requirements. First, bacterial retention testing is crucial. It verifies that our filters effectively remove bacteria, ensuring the sterility of the final product. Next, we conduct product compatibility studies to confirm that the filtration process doesn't negatively impact the product itself. We also perform integrity test correlation, which helps us verify that the integrity tests we use are reliable indicators of filter performance. Flow rate and pressure evaluation are important as well, ensuring that the filtration process runs efficiently without compromising quality. Additionally, extractables and leachables studies are essential to identify any potential contaminants that could be released from the filter materials into the product. Chemical compatibility testing further confirms that the filter materials are compatible with the product, preventing any adverse reactions. Lastly, adsorption assessment evaluates whether the filter might unintentionally adsorb or remove the active components of our product. The overarching objective here is to demonstrate that our filtration process consistently produces sterile filtrate without affecting the quality of the product. These validation activities together form a comprehensive approach, ensuring reliability, safety, and product integrity throughout the process. Now, let's take a look at the Brevin de Mona's diminuta challenge test, which is a crucial part of our validation method. In this test, we challenge the filters with Brevin de Mona's diminuta, a microbe known for its small size and ability to test sterilizing filters effectively. Typically, we use a challenge level of at least 10 to the 7th CFU per square centimeter of filter area. This ensures we're testing under robust, worst-case conditions, pushing the filter to its limits. We also simulate the maximum process duration and pressure that the filter might experience in real-world applications. This way, we can confidently assess its performance under demanding conditions. The key acceptance criterion: no bacterial penetration through the filter. If the filter can prevent bacteria from passing through under these challenging conditions, it demonstrates reliable sterilizing capability. This test provides essential scientific evidence that our filters are effective at sterilization, which ties back to our overall goal of ensuring safety and quality in our processes. Let's take a look at how we verify filter performance, which is a critical part of ensuring product safety and quality. We use common integrity tests like the bubble point test, diffusion test, and pressure hold test. Each of these helps us confirm that the filter is functioning properly and hasn't been compromised. The bubble point test, for example, checks the pore size and integrity by seeing at what pressure air bubbles start to pass through the filter. The diffusion test measures how well the filter restricts the passage of gases or liquids, providing insight into its integrity. And the pressure hold test involves applying pressure to ensure the filter can withstand operational conditions without leaks. Now, from a regulatory perspective, particularly in the EU GMP Annex 1, there's a strong emphasis on pre-use and post-use integrity testing, often referred to as PUPSIT. These tests are important because they help us verify that the filter remains intact throughout the manufacturing process. Another key point is that the integrity tests we perform must correlate with the filter's ability to retain bacteria. This means that passing the integrity test is a good indicator that the filter will effectively prevent microbial contamination. The main objective here is to confirm the filter's integrity both before we start manufacturing and after we've completed our operations. This ensures that at all times, the filtration system is reliable and effective, safeguarding the quality of our products and compliance with regulations. Now, let's take a closer look at bioburden and process controls. First, when it comes to bioburden control measures, maintaining a controlled manufacturing environment is absolutely crucial. This includes ensuring that all equipment and pipelines are thoroughly sanitized to prevent any microbial contamination. Timely filtration right after solution preparation is also key, as it helps catch any microbes early on. Additionally, where applicable, using prefiltration systems can further reduce bioburden, adding an extra layer of protection. Moving on to process monitoring parameters, it's important to keep an eye on several critical factors. Differential pressure helps us monitor filter integrity and detect any potential clogging. Flow rate ensures that the process runs smoothly without stressing the system. Filtration time gives us insights into process efficiency, and temperature control is vital because it affects microbial growth and filtration performance. Finally, the regulatory focus here is on minimizing microbial load before the sterilizing filtration step. This is a fundamental aspect of ensuring product safety and compliance. By effectively controlling bioburden early in the process, we help safeguard the final product's quality and meet regulatory standards. All these measures work together to create a robust, reliable process that prioritizes safety at every stage. Now, let's talk about maintaining a validated state in our filtration systems. It's crucial that we regularly assess any changes that could impact performance. For example, any supplier changes for filters need to be carefully evaluated to ensure they meet our quality standards. Similarly, if there are any changes in membrane materials, we must verify that these modifications don't compromise the filtration process. Process parameter adjustments also require thorough assessment. Even small tweaks can have significant effects, so we need to be vigilant. When it comes to product formulation changes, these can alter the filtration requirements, making revalidation essential. Equipment modifications, too, must be scrutinized to confirm they don't introduce new risks or deviations. Beyond managing changes, lifecycle activities are key to maintaining validation. This includes periodic reviews and revalidation to confirm the system remains effective over time. We should also be trending filtration failures and deviations, as these insights help us identify patterns and prevent issues before they escalate. Implementing corrective and preventive actions, or CAPA, is vital to address root causes and continuously improve. The main objective here is to ensure our filtration remains effective and compliant throughout its life cycle. By staying proactive and diligent, we can uphold the integrity of our processes and product quality at all times. Let's take a closer look at some of the common regulatory observations we often encounter. These are frequent deficiencies that can lead to serious regulatory issues if not properly addressed. For instance, inadequate filter validation documentation is a key concern because it impacts our ability to demonstrate that filters are effective and reliable. Missing integrity testing records is another critical point. Without proper records, it's difficult to prove that filters have maintained their integrity during use. Incomplete risk assessments can leave gaps in understanding potential hazards, which is why thorough evaluation is essential. Weak bioburden monitoring is also a common problem, as improper monitoring can compromise product safety. Inadequate change control procedures pose risks because any modifications to processes or equipment need to be carefully managed to maintain compliance. Lastly, lacking filter compatibility studies can result in unforeseen issues during manufacturing, affecting product quality. Now, let's consider the potential consequences of these deficiencies. The FDA can issue warning letters, which are formal notices that highlight noncompliance and require prompt corrective actions. These issues can also jeopardize sterility assurance, putting patient safety at risk. In some cases, products might need to be recalled, which is costly and damaging to reputation. Additionally, regulatory inspections often reveal these kinds of deficiencies, leading to further scrutiny and potential penalties. Connecting this back to our overall theme, it's clear that proactively addressing these common deficiencies is vital not only for compliance, but also for ensuring the safety and efficacy of our products. When it comes to best practices for compliance, strengthening our sterilizing filtration systems is absolutely key. We want to implement a risk-based filtration strategy, which means assessing where the greatest risks are and tailoring our filtration approach accordingly. It's crucial to use validated sterilizing-grade filters to ensure they meet the necessary standards for safety and effectiveness. Routine integrity testing is also essential. This helps us verify that our filters are functioning properly over time. Maintaining robust bioburden control measures is vital to prevent contamination before it can happen. Periodic revalidation of our filtration processes ensures that everything remains effective as conditions change. Training personnel thoroughly on aseptic filtration practices cannot be overstated. Well-trained staff are our first line of defense against contamination. We should also integrate filtration into our overall contamination control strategy, making sure it's part of a comprehensive approach rather than an isolated step. And of course, maintaining complete documentation and traceability is fundamental for compliance and continuous improvement. Now, moving into continuous improvement, it's important to monitor deviations and filtration failures closely. By tracking these issues, we can identify trends and areas for enhancement. Keeping an eye on process capability and filter performance helps us ensure consistent quality. Additionally, reviewing emerging regulatory guidance keeps us aligned with the latest standards and best practices. All of these steps together help us strengthen our sterilizing filtration systems and ensure ongoing compliance, which ultimately supports the integrity of our products and the safety of our patients. So, in conclusion, 0.2 micron sterilizing filters are truly essential for maintaining sterility in pharmaceutical manufacturing. They play a critical role in ensuring that products are safe and free from microbial contamination. Risk assessment and validation are key components here, because they help us ensure that these filters reliably retain microbes and that our entire process performs as expected. It's about building confidence in our sterilization methods. Regulatory compliance is also a major aspect. To meet these standards, we need solid scientific justification, proper integrity testing, and effective lifecycle management of our filtration systems. These steps demonstrate that we are consistently controlling bioburden and maintaining product safety. Effective bioburden control and change management further strengthen our sterility assurance. By actively managing changes and controlling microbial load, we reduce risks and maintain high-quality standards. Finally, robust filtration systems support not just patient safety and product quality, but also help us stay inspection-ready. They form the backbone of a reliable, sterile manufacturing process, reinforcing our commitment to delivering safe, effective medicines. For further support, please don't hesitate to reach out. We're here to help you with any questions or additional guidance you might need. Remember, your success is our priority, so feel free to contact us whenever you need assistance. Thank you for your time and engagement today. We truly appreciate your attention and look forward to supporting you in your journey.